Welcome to NOAA Live Alaska. My name is Lisa Hebrew-Key-Raring, and I'm going to be moderating today's webinar. This series is a collaborative effort by NOAA Fisheries Alaska Fisheries Science Center, where I work, NOAA's Alaska Regional Collaboration Network, and NOAA's National Weather Service. This webinar series is designed to help you get to know NOAA's work in Alaska and how we connect and work with your communities. NOAA, the National Oceanic and Atmospheric Administration, studies the ocean and the atmosphere and where the two meet, from weather to ocean to the animals that live around us. All of our speakers work for some part of NOAA or work in partnership with NOAA. We hope this gives you a sneak peek at the different kinds of careers that you might be interested in. Today, we're introducing you to Coowe Walker and Jacob Argueta, who work for NOAA's Kachemak Bay National Estuarine Research Reserve in Kachemak Bay, Alaska. While we'll be talking about NOAA's role in research and stewardship, we want to recognize that we're all coming to you from the traditional lands of Native communities who have substantial traditional and local knowledge and much to share with us. We'd like to also acknowledge that Coowe and Jacob are presenting from the land of the Dena'ina and Sugpiaq people, and we're hosting this webinar from the traditional lands of the first people of Seattle, the Duwamish people past and present. A few guidelines before I hand you over to our speaker. You're all muted because we have a lot of people on the line and we want to make sure that everyone can hear our speakers. However, there's a box where you can write questions. We encourage you to ask them as we go, and my colleague Chris Beyer and I will be keeping track behind the scenes for Coowe and Jacob. They'll stop every now and again and answer a few questions. We might not get to all of our questions, but we'll try to answer as many as we can. Now, I'm going to try to get the slide back to our first title slide so that you can see what our talk is about, salmon in winter, and then I will hand it over to Coowe and Jacob to introduce themselves. So you guys can go ahead and unmute yourselves if you're muted.
Thanks, Lisa. It's really nice to be here. I'm Coowe Walker. I'm the manager and watershed ecologist, or lead watershed ecologist for the Kachemak Bay National Estuarine Research Reserve, located in South Central Alaska on beautiful Kachemak Bay. Just a little bit about us. They asked us to share a little about our personal background. So here's a picture on the left of myself. I think I was about 5 years old. When I was a kid, we traveled a lot, and we traveled by boat. And so we took everything with us in these trunks that I'm lying on. And we lived overseas quite a bit. And I really liked it in a lot of ways. Seeing new places, traveling with my family, but it also made me kind of nervous. And one of the things that grounded me was my interest in the natural world. So I knew from a pretty young age that I wanted to study natural systems and how natural systems are connected and how we're connected to them. So in the middle there, I went to college, and my first year in college, I got an internship. And that's me in the middle. And my first day of my internship, my parents drove me up, and that's my mentor, Carol, on the right there. Important person in my life then and still now. We've maintained a connection over the years. And then on the right is me and my chosen habitat, which is streams on the Kenai Peninsula. I love working in streams and understanding how streams function and the fish that are in them. And that's what I've been doing for the past 20 years, and we're going to share more about some of the aspects of that work. Over to you, Jacob. Next slide, please.
Hi, everybody. My name's Jacob Argueta. I also work with Coowe here at the Kachemak Bay Research Reserve in Homer, Alaska. Originally, I'm from outside of Portland, Oregon. I grew up on a farm down there, and spending a lot of time outside around animals and growing vegetables with my folks, I think really instilled a love for being outside and learning through doing and getting my hands dirty. And as I grew up, that kind of transitioned into a love of kind of being sustained by being outside. In that middle picture, you can see me with a mushroom that we foraged. That's a king bolete, a delicious edible mushroom. And from kind of getting that sustenance from nature, it also led to a desire to sustain nature in my career. So in college, I studied environmental science and geography, specifically geographic information systems, and was fortunate enough to get an internship with the reserve while I was an undergraduate. And then when I graduated, I came back up for a second internship, which landed me in a technician position after summer. And I've been working with the reserve for about almost 4 years now. And I've again, lots of feelings of being fortunate, but yeah, just feel very grateful that I get to spend my work days outside a lot of the time doing work that feels very meaningful and important to me. And I get to do a lot of fun stuff outside. Like here on the right, I'm flying a drone, which we use to collect imagery and data in a lot of our projects. But other fun stuff, like Coowe was in that stream, we get to splash around in the water a lot, taking water samples and water measurements, as well as catching fish in the near shore, and collecting data all over the place. So it's a great time being out here.
Awesome. I love how happy you look in that photo, Jacob. Well, I'd like to take a moment before we dig in to just acknowledge that we are coming to you, both Jacob and I, from Kachemak Bay, which is the borderlands of the Dena'ina and Suspiaq peoples. And they have taken care of this land for thousands of years and continue to help us understand how to steward in the present. And without that stewardship, we would not be able to do the work that we're doing. So we're very appreciative and thankful for that. Next slide, please. So we are one of the National Estuarine Research Reserve System, research reserves in the system. That's a big mouthful. We can just say NER for short. And you can see that there are a number of reserves all around the country. There's 29 in all. We're the only one in Alaska and the largest one by far. We partner, so it's a NOAA system, NOAA-led system that partners with a state entity. And in our case, we're really fortunate to be partnered with the University of Alaska, Anchorage, the Alaska Center for Conservation Science. Next slide, please. And here you get a little closer look of where we are in Alaska. This is Kachemak Bay, showing you the official boundary of our reserve in yellow there. So it includes the critical habitat area of Kachemak Bay, the waters of Kachemak Bay, and the Fox River Flats at the head of Kachemak Bay. And then this on the south, where you see Lisa's cursor there, that's the Kachemak Bay State Park. So it includes all these official boundaries, but we also work beyond those boundaries. And if you look to the north of the bay and you see the blue lines there, the river, that's the Anchor River, and we work a lot in that river, and you're going to see a lot of pictures from that river, because those rivers are where the salmon go when they're not in the ocean. Next slide, please. Okay, so we do a lot of research in the summertime. We love working with students. There's several interns in this photo. We look at salmon in these, this is an estuary system, and we ask a lot of questions about what the salmon are doing there, how they're growing, where they're moving, things like that. And as you'll see, we also work later in the winter. But first, I think Jacob is going to introduce you to summertime research through a video.
Yeah, so real quick before we jump into this video, this is a method of catching fish called electro fishing. And it's important to note that this is just stunning the fish. It's not permanently harming them in any way. And we can jump right into that video and I'll kind of explain what we're doing in it.
Okay, I'm just setting up the video right now. It's a little bit slow, but here we go. Get it. Don't forget that you need to unmute yourself.
Here we go. As we start up, we're walking out to one of our sampling sites. And as we're getting there, of course, we need to gear up. A big part of our gear is waders, so making sure everybody's set and fit. And now these are my two assistants, Courtney and Daisy, and I'm passing them some nets. They're going to be following behind me as I'm electrofishing. So there I am setting up the electrofishing backpack. That is basically a battery that's extended with an anode and a cathode end. That ring right there that you see and that little wire that I just moved. So I put those in the water, and I'm able to put in a small electrical current, and that stuns the baby fish, and Daisy and Courtney behind me will be able to scoop them up as they float to the surface. They're getting nice and set. That backpack is pretty heavy, so having it nice and secure is always good. This is the landscape around the streams we often find this tall grass. Making sure everybody's set. You can note that they're wearing rubber gloves to, again, protect them from that electric current. And there's a shot of what we're after, juvenile salmon. Here we see the stream. It's kind of murky this time of year. It's next to a peatland, so there's a lot of organic matter being leached into this stream, giving it that dark color. Here we see a quick peek under the water at these guys. There they are. Important to note that white line on the top of their dorsal fin and their anal fin as well. It's a very clear indicator that they're probably going to be juvenile coho salmon. Here we go, getting in the water. I'm up ahead with the Electro Fisher. And like I said, Courtney and Daisy behind me, scooping any fish they see that are floating to the surface after I briefly stun them with the electric current. It's important to check our nets every now and then to make sure we're getting them out of the water and not continuously shocking them while they're in the nets. There we go. See, they're right back to jumping around even after being stunned for a sec. We store them in those buckets until we've completed our electro fishing session, and then we can take a look at them. You can see I'm kind of reaching under those banks. In these systems, the stream banks are often undercut a lot, so they can be almost feet to either side of the stream there of undercut. And the salmon like to hide out underneath of those. Here we are getting out. I think that's Coowe picking some invertebrates out of the net as well. And we use these photariums, these little clear viewing windows, to get a better look at the fish, as well as take some measurements like length there. You can see along the bottom. We can estimate their length in millimeters without having to take them out of the water. Yeah, and those are some beautiful little coho salmon. And once we're done taking a look at them, we put them right back where they came from. That's a quick little snapshot of what catching juvenile salmon looks like using an electro fisher. I think we might have time for some questions.
Great. So I actually had a question that came up as we were looking at the stream. It looked like the stream was really narrow and deep. Is that because you were saying that it undercuts the banks a lot? Are most streams around Kachemak Bay like that?
So in our area, it's a pretty flat topography. So a lot of these headwater streams are relatively slow moving. And as they cross through, that area specifically is near peatlands, specific type of wetland. And it really slows down the flow and creates those kind of deep channels like we were seeing. I don't know if you want to add anything to that, Coowe.
Well, it's really easily, the peat material's easily eroded, like cut through by the water. So in the spring, when we get really big floods, it actually overbanks that stream area, and then as it recedes, it cuts down too. So over time, it creates this, what we say, incised, so like a deeper channel, but it's not always, the water isn't always that low. It changes quite a bit. It can be up and over the banks, and usually is in the spring. And sometimes, yeah, when the snow melts.
Sorry, I was just going to remind our viewers that if you have any questions about the video that you just saw of the sampling that they were doing with the baby salmon, please write your questions in. There was a question about how many baby salmon do you usually collect in a survey like that?
That's a good question. It's pretty site specific, but that was a pretty short bit of stream that we were sampling there.
Do you want to answer that?
Maybe 50 meters, and we caught a good 10 salmon. To accurately estimate the actual density in an area, we would block off the upper and lower limits of that stream and do multiple passes to get a good idea of what the actual population of that area looks like. But short answer, it varies.
And then what kind of information do you get from the salmon when you collect them like that?
Lots of different stuff. So we go into further detail later on in the talk about some of the things we learn in these surveys, but a couple quick answers are age, diet, species, stuff like that. It's pretty common to learn about them.
Okay, sounds great.
How
Well,
big they are.
And how big they are, great. So if you guys have any other questions, please type them into the question box, and then maybe we'll go into the next section and answer any other questions after that.
Yeah. So what we move into next, we're going to think about salmon life history. That means what they do throughout their lifetimes. And we use a word called anadromous for that. Have people heard of the word anadromous? Can raise your hand or put it in the chat.
If you've heard the word anadromous, or if you know what it means, you can type your answer into the chat box or into the question box, and we'll read out some of the answers. And let's see, we don't have a lot of answers to the anadromous question yet, but we did get a couple of questions. Maybe we can handle these couple of questions while people are thinking about anadromous. Clary from Nome was asking, how deep is the water in the streams that you were looking at?
So I'm about 6 feet 1 inches tall, and that water was coming up to my waist, so maybe 3 to 3.5 feet, 4 feet.
So if you fell in the water, it would be like a shallow end of a swimming pool or something like that.
Yeah, plus the drop from the bank is a bit steep too, so you don't want to be tripping into these streams.
And then one more question before we get answers about what anadromous is Stephanie was asking, is the goal to collect all the fish in a particular survey area or just a sampling of it?
It's going to be pretty hard to collect all the fish in a system, so it's usually a subsample. But unless we're doing something like I said before, where you block off what we call a reach, so a section of the stream, and then we're trying to catch all the fish in that specific small subsection of the stream.
Gotcha.
So we have, Stephanie, we have different goals. Sometimes we're looking for, we have one goal, sometimes we have another. Sometimes we just want to show people, like fishermen or people that live near that stream, that baby salmon are living there. And then we just go in, like you saw in the video, and catch fish. Other times, like Jacob was saying, we're actually trying to quantify or figure out, an estimate of how many fish are in a certain reach. And we do that in all different systems, and we can compare what types of habitat and how many fish are in each different habitat. And then sometimes we just want to collect fish so that we can say we want to measure their growth, or we want to see how old they are, and we might just go in and sample a few fish. So it really depends on what what the goal is. We do a lot of different kinds of projects.
Great. Well, we have a couple of answers for your question about anadromous. Texas from Colorado was thinking that anadromous means that the fish live in both the ocean and the rivers. And Michelle from Hawaii was saying that fish can live in both fresh and salt water.
Yes, you guys are right. So if you look at this picture here, this is a drawing, an illustration that was actually done by an Alaskan youth. So it's like some of the folks on our call, and it's showing you the life cycle of Pacific salmon. And you can see there's the ocean bubble in the lower part and the river bubble in the upper part. And both adults and juveniles use both the river and the ocean at different times. And in the middle, you see the estuary. That's where the ocean meets the river, and we have a mixture of river and ocean conditions. So when you're in the middle there, salmon use that estuary in a variety of ways, and you'll learn a little bit more about that. And they also use it to go through what we call smoltification, which means they're getting ready to transition into being an ocean fish. And that's where they spend their adult years before they return, and they spawn in the rivers, and their eggs hatch, and then they emerge and they become juveniles. And depending on the species, they may spend, in our region, 1 to 3 or even maybe 4 years in the freshwater before they go back out to the ocean. So let's see, let's move on to the next slide. This, I think folks from Alaska at least probably know that we have a number of different species of Pacific salmon and also trout, and each one has their own identifying characteristics. We're going to focus on coho salmon, which is where you see the circle there on the left. And just as you look at this fish here that's Lisa's circling, note how it has kind of a relatively deep body, so it's kind of, you know, deeper or wider top to bottom than some of the other species. And also look at that eye. That's a big eye. And then if you look at the fins, the anal fin and the dorsal fin, you can see a white line. Okay, so now we're going to go to the next slide and let's see if you can see some of those features on this real fish in the picture. There it is. I love seeing these baby fish.
I can see that big eye. So here's the big eye here. And I can see that white line. It really stands out, the white line on the dorsal fin and the anal fin here.
Yeah, sometimes those, you know, they can be tricky because as they live in the stream, those fins get kind of brushed up against the bottom or the top of the bank if they're in an undercut, and those white lines can rub off. So it can be tricky to tell the species apart sometimes. We spend a lot of time sometimes trying to figure out whether it's a coho or a chinook, but when you have one this clear, you know for sure. And I'd like to ask everybody on the call if you've ever seen a baby salmon, either in the summer, maybe in the winter, maybe even in your classroom.
So if you've seen a baby salmon, please write into the chat box whether you've seen one, and if you have seen a different, what species it is, if you know. We have Texas is saying no. Idan is saying no. Cheryl is saying 4. Oh, 4 out of 14 people in Mrs. Williams' class in Palmer, Alaska. Michelle is saying yes, she's seen them. Angela is saying no. Mitsuko has said I've seen several. So it looks like, and Eve and Jasper have been saying yes. So it looks like we've got a mix of people. Some have seen baby salmon and some have not.
Did anybody say where they'd seen them?
Sorry, I need to unmute myself. I
Oh.
keep forgetting. So, yes. So, Clary from Nome is saying, yes, I saw a pink baby salmon. Cecilia was saying, I've seen some in Potter Marsh in Anchorage. Pam from Tyonek is saying that they have a salmon project there, so that might be a salmon in the classroom. And Mitsuko is saying that she saw baby salmon at a hatchery. And Mrs. Williams is saying that they saw baby salmon at Matanuska Lake. And Michelle was saying that she saw baby salmon in Seattle. We've seen salmon at the Ballard Locks here in Seattle as well. Westchester Lagoon, also in Anchorage. That's Cecilia saying that. And Pam from Tyonek is saying that they currently have alevin. I never know how to pronounce that word. Is it alevin or alevin? I never know.
I say Elvin, but I think people have, I don't think there's a single correct way. Maybe I'm wrong, but we know what you mean.
And let's see. So Clary was saying that she has seen it in Pilgrim. I don't know whether that's a river. And Eve says that there's a hatchery in Juneau where they see baby salmon. Mrs. Williams is saying that some of her students have seen them at the Sea Life Center. And Idan is saying, I think Idan is from Germany, and he was saying that they were saying that there was no salmon there. So something new. And then Eve is also saying, Jasper are also saying they see salmon by the Mendenhall Glacier in Juneau.
Oh, that's really fun to hear where people have seen baby salmon. It's great that you guys have seen them too. You'd be surprised how many people who depend on salmon for their livelihoods and their sustenance have never seen a baby salmon. So next we're going to delve into how we know where baby salmon are, now that we've heard a little bit about where you've seen them. And if you go to the next slide, please, we're going to talk a bit about this slide, sorry. This is the right one. Finding and tracking. Yeah,
I was trying to
that's
figure
good.
out where oh, sorry.
Go.
I
That's
seem to
okay.
have
Just go
gone
back.
into the wrong, yeah, there.
Yeah. Okay. So we're going to talk about how we find them and how we use instruments to track them. So next slide, please. These are a couple of ways that we use. You can see on the left there, it's what we call it, you might have heard people say a PIT tag or a passive integrated transponder tag. They're very tiny, like I think Jacob's going to show you one a little later. And we put them in the abdomen, right under the in the abdominal cavity of the fish. You can see there on the, yeah, right there. Yep. And I think of it as kind of the equivalent of, you know, by size, if you took a soda can and put it in my abdominal cavity, it sounds big. It is big compared to the fish, but they tolerate them very well. And we've been able to track fish over, you know, several years, which is a really powerful tool so that we can understand how that individual fish is moving and how it's growing. And then on the right, you see a different type of tracker. This is we call it VIE or visual implant elastometer. They're basically color coded. They come in many different colors, and you can tag a fish in many different locations. And using combinations of colors and locations, you can identify individual fish, and then you go back and try and recapture that same fish. This is a great tool. It's not quite as you know, cumbersome for the fish. However, they do wear out as the fish grows. They basically grow over, like their skin grows over the colors, and so they don't last very long. The pit tags last a really long time. Okay, next, I think Jacob is going to show you some of this gear live.
Yeah, so real quick, I'm just gonna show everybody some of those tools that Coowe was talking about. So with me, I have a pit tagger. So this is kind of the instrument they use to insert that little guy. On the end is a hypodermic needle, and using my prop Coho, I'm not gonna actually put it in, but what you would do is you would wanna get him on his belly, and you're injecting that pit tag into its abdominal cavity. And I'm going to squeeze it out. So you squeeze the trigger. Whoop, there it goes. And we see a pit tag. So each one of these has an individual number associated with it. And what we can do is use a scanner like this guy that reads the individual number, and we can identify individual fish. These pit tags are also similar to what you would find in your pet if they've been chipped. That's what a pit tag is similar to a chipped pet. And that lives inside of the little salmon, the little pit tag, as you can see. And that's about the right size difference there, about that size to this size fish. That's the pit tag.
I love the little felt fish. Great, thanks, Jacob. Well, now we can go to the next slide, and you can see.
All right, let's see whether I can get this there.
Okay, so these are antennas. So this is what we put in the water. So the way the tag works is when it goes through, you can see that PVC, that white outline. That's a pass-through area. And the other things that are hanging down are actually made of conveyor belt. They're big rubber, and that's basically what we're trying to do is get the fish to pass through a certain area, because then the antenna reads the sign. So Jacob showed you a handheld antenna. These antennas live in the stream as long as we want them to, and so we can see how they move over long periods of time. And you might be wondering how the fish gets to that hole because it's above the water line. This is in an estuary, so the tide's coming up and down, and when the tide comes up, the fish are able to move into this marshy area, and they go through that hole, and then we can see where they're going. So you might be wondering, or I would think you might be wondering, why do you have two? Why are there two antennas right next to each other? Does anybody have any ideas?
Are the antennas these things here that are sticking
anten-
up, or are the
No, so
antennas in this framework?
the antennas are inside that PVC pipe.
Right.
Yeah, it's a pretty high tech system that this one you can build them yourself. We actually had this one built by a local engineer who helped us out, but it's inside of that PVC pipe, and then it's connected to a power source and what we call a reader that collects the information.
So there's one antenna on this side, on the side of the wooden thing that we're seeing here, and then there's an antenna on the other side. Is that what you're saying?
No, so right where your cursor is, that's one antenna, and then go to the back of the picture, and you see that structure in the stream.
Oh, this one.
That's another antenna. So why
Gotcha.
would we think we would want to have two in the same place?
So why do you think that we would have why Coowe and Jacob would have two antennas, one here and one here? Why do you think that they would want to know which fish are going through and have two antennas so close together? Cecilia is wondering whether it's to measure the speed of the fish swimming. Idan is saying that salmon can jump, so maybe they're thinking that they might jump over one of the antennas and get to the next one.
Those are
Yeah, so what do you think, Coowe?
so yeah, those are so actually both of those things we work with. The jumping is why the antennas are so high and why we have all those baffles, we call them, to keep the fish from kind of channel them through the antenna. And the speed is that is actually one of the things we can measure. What we're doing is we're trying to figure out direction, and from that we can know speed too, like how fast. So when it goes through one antenna, we get bing, and we see a number pop up that's fish. And then it goes through the next antenna, bing, we know that fish went through that antenna too. So we know how fast it went, and we know which direction it was going. If it hit one antenna first and then the second, we know it was going, say, up or down. So that's really helpful. And if you go to the next slide, you can see some of these patterns. And you can also see there's on the left, you see the antenna, and you also see that I mentioned that we have a power and reader system. That's what's in that tall box there. And then on the right, you see some of these patterns from some of the fish. So you can see some of the fish are moving up, some of the fish are moving down. Sometimes they move up and down in the same day. You're looking down at an estuary. You can see the channels. The ocean is the tide is out, so it's that kind of, it just looks like a mud flat right now, but basically the ocean would be on the left there, and the estuary is on the right where the fish are moving around. And I'd like to ask folks if they have an idea of why do you think they move so much?
So what you're seeing on the map, folks, is that the yellow arrows that you see are the downstream movement. So Coowe was saying from the previous slide that they can tell which direction the baby salmon are swimming because of which order they go through those two transmitters. And then the blue arrows are upstream movement. So why do you think that they have to move around quite as much, or why would they want to move around that much in the estuary area? Pam is saying to get oxygen. Idan is saying to keep alive. Let's see. I was wondering whether there are conditions like time of day that makes a difference. Ola is saying changing life stages. And I was actually wondering, Coowe, whether these movements that you were looking at, what length of time is that over? Is that over a couple of days, or is that over a couple of weeks?
So in this slide is actually showing representative fish. We collect thousands of data points, but I can tell you that some fish move in a daytime. Will move all the way from the mouth of the river all the way back up to the tree line. Other fish will stay put and just move kind of like they'll move just within a small range. Some fish go to the river and then with the tide go back up in the channels and then go back out again. Some fish just come down river and move right out to the ocean. So they do a whole bunch of different things. Kind of like people do a whole bunch of different things in their lives, but all of them are using the habitat. And what's really remarkable about salmon is that they do so many different things. They use their habitat in so many different ways. It makes it complex. It's really interesting to study them because they're very diverse. They do a lot of different things. But by doing all those different things, it means that at least some of them are going to do the right thing in any one year to survive. And that's what makes salmon so resilient and remarkable. So if you go to the next slide, we have a kind of a question for you, like, what do salmon need to survive? Shelter, water, food, space, or all of the above?
So just so that we're so was the answer to your previous question of why do they move around so much, is that because of habitat?
Yes, thank you for helping me clarify that. Yes, they use different types of habitat. Individuals use the habitats in different ways, and overall they use different habitats at different times of their life.
Gotcha. And so we've been getting a lot of answers for your question here. Texas, Mitsuko, Jody, Idan, Mrs. Williams' class are saying E. Ola is also saying E, all of the above. Clary is saying water is important. But most people, I think, are saying that all of the above, the shelter, water, food, space, all of those things are important in habitat.
Yay, I'm so glad you said that. Yes, good thumbs up. So we're going to focus in on, now we're going to kind of shift into the winter habitat. And what does habitat look like for a salmon in winter? This is generally oh yeah,
I was
sorry,
wondering
I just want to
whether, just before we go into the winter habitat, whether you have enough time for a couple of questions.
Sure, it's up to you guys. I have plenty of time, but
Because I think there were a couple of questions that came up from when Jacob was showing the pit tags. So Jasper from Juneau had wanted to know what happens if a fish has a tracker and it dies. Do you know when it disappears, or what happens?
So really, we are only seeing those the information from the tagged fish come up when they pass through either one of those antennas like Coowe was showing, or if we recapture them in either a seine or possibly through electro fishing and scan them again with like the handheld readers and identify those individual fish. There's not really a way of knowing if the fish dies just because it's not continuously tracking it. We only get the information from it when it passes one of the sensors. I don't know,
the
Coowe, have
only
you ever
way,
seen an adult come back with a pit tag in it?
oh, adult, sorry, I was thinking of the sometimes I'll answer from a juvenile perspective. Sometimes they do die, and the only way we can tell is that they just when they die, they'll just kind of be in the same place and for a really long time, and then we're like, oh, okay, either that tag fell out or that fish died. So we can tell from the data somewhat. But no, we have not monitored for adult fish coming back. It would be, yeah, we haven't yet. It would be really interesting to do that. We'd have to tag a huge number of fish because you guys probably know this. I'm thinking maybe that, the majority of baby salmon that go out to the sea don't survive. So you'd have to tag a whole bunch of them in order to get, a few adult fish back. So we haven't done it yet. We've been focusing on what the juveniles are doing in the watershed areas.
And I think that probably answered Kip's question, because Kip had asked, are pit tags recovered, or might a person find one in a slab of salmon they purchase at the store? And so you just answered that only a few of those many, many, many salmon that become smolts and baby salmon grow up to become adults, because there's so many different predators out there and sources of mortality, right? So it'd probably be pretty incredible to find a pit tag in a slab of salmon at the store.
Yeah, that would be pretty exciting, but I haven't heard of that happening.
All right, well, let's move on to the winter habitat then of the salmon.
So I think
let's see what
Jacob's gonna, while you get the video going, just turn it over to Jacob, who's gonna kind of help us get immersed in the wintertime habitat.
Yeah, if you would want to just go ahead and play that, I can give a little talk on where we are.
Don't forget to unmute.
Really quick, we're just seeing a clip of a stream in winter. This is somewhat typical of a larger end stream that's not completely frozen over. It's somewhat fast moving area. This is actually from just outside the Tabona School in Tyonek. So if you're tuning in from Tyonek, you probably recognize this stream, and it probably, I'm imagining, looks somewhat similar right now. And yeah, it looks cold. So we're going to see what the juvenile salmon are doing at this time of year.
Awesome. So let's see the next slide. We'll just give you a still life preview of what it looks like in the winter and what it looks like doing research in the winter with those minnow traps. And I think you'll get another opportunity to see those the equipment a little bit later. Can we go to the next slide, please? Okay, so in the winter, things are a little bit different than the summer in terms of how we prepare ourselves because it's cold, right? So you see on the left here, it's one of our past interns, Anna, doing summertime research, and she's got her baseball cap on and putting her waders on and looking pretty comfortable in the green all around, just a sweatshirt, hoodie maybe. And then you can see on the right here, I think this is Serena, who's our education coordinator, helping out in the field, and she's got her woolly hat on and her coat and her boots and her thermal underwear, I'm sure, too. So just quite a different experience to be doing work in the wintertime. It's a lot harder. And let's see, we'll go to the next slide, please.
Are you seeing the next slide? I just changed it.
Oh, yes, sorry. I thought you heard me. Oh, there we go. So this is an open water lead that's kept open by groundwater, and you can see how it's snowy all around. And if you look at the slide, you see the open water, and then you can see kind of it extends in the distance. It's a big overwintering area that we just found by walking around, by finding, basically looking for off-channel habitats where salmon might be in the wintertime. Next slide, please. Here's a little bit closer view of that. This is looking right up at this groundwater seep as it's coming in. It's keeping, you can see the open water where it's snow and ice all around. And what we're going to talk about now is how do we catch the fish under the ice? And Jacob's going to, we're going to play a couple of videos of how we're trapping under the ice. And then Jacob's going to show you some of the equipment in lifetime.
So
let's see, we're gonna have a video of under the ice.
All right.
Yeah, let's take a look at that one.
So
So again, this is that same stream in Tyonek. Oftentimes, we're dealing with places where the ice is coming in a bit more. This is kind of just a peek of the ice forming on the sides of the channel and kind of what the conditions would be like if you're a juvenile fish under there. There's some good flow, there's some good woody debris that probably is good little hiding spots. But again, it is cold out. I think ice was forming on the lens of that camera right after it came out.
all right, and I'll start the Minnow Trap one now.
Yeah.
So this is how you what you use to catch the
Right, so we're seeing a lot more ice formed in this one, and compared to the summer where it's kind of easy to walk through the stream with, say, an electro fisher, in the winter when there's lots of ice, it's hard to use a technique like that. So we often use what are called minnow traps, which is what we're seeing right here, and I'm going to show you one in person in a second. I'm not entirely sure why they call them minnow traps, but I can imagine that it's because they catch minnow-sized fish, in this case, juvenile salmon.
All right, so did you want to show the Minnow Trap now, or?
Yeah, I can do that. So this is the same kind that you just saw in the middle in the video. This one's all folded up. We have them tied up with this nice bright green line so we can tie it to the bank so they don't float away. And we can find them again easily just based on that. And I'm doing the clips on the side. Here we go, and it expands kind of like an accordion almost. I can get this guy off. This one's been sitting for a second. There we go. And boom, it pops on up. So the way these guys work is it sits in the water, and there's a little slot in the top here with a pocket. You can see my fingers in there, the pocket, where we can put bait. And as it sits in the water, fish are attracted to that smell, and they can swim in these openings on either end. Bring back my prop. It's just about the right size for a little juvenile salmon to swim on in there. And then once they're in, since the opening is so small, it's actually pretty hard for them to get back out. So they're kind of trapped in there until we come. And imagine this is underwater, and we can just pull it on up by the string and check and see what we got in there. And that's an easy way to sample for juvenile salmon under the ice.
That's great. We did get some questions about how big can the salmon get when you're trapping them under the ice?
That is a good question. So like Coowe mentioned, the salmon in the area can spend multiple years in fresh water before they're heading out to the ocean. So you're seeing them anywhere from, I don't know, just a couple millimeters, maybe like 14 millimeters all the way on up to a couple-year-old fish, which are reaching 70, 80 millimeters, so quite a bit bigger than that. And oftentimes, depending on where we're fishing, you can see other species of fish too, like sculpin or sticklebacks ending up in those minnow traps too.
Well, that's very interesting. I don't think that I realized that salmon spend that much time in the estuary. Is that typical of most habitats, or is it only in areas where there's a lot of marsh, a lot of area where they can spend, that time safely?
There's a lot of yeah, sure. There's a lot of variability, Lisa. So it's not just the estuary, they live all the way in the tiny headwaters, like where you saw that Electro Fishing video way back. That's high up in the river. So they live 1 to 3 years, or sometimes 4, and it varies by where you are. So down where you are in Seattle, they spend less time. These are coho salmon, so it varies by species too. Pink salmon don't spend much time at all. They just pretty much hatch and go back out to the ocean. Chinook or king salmon spend at least a couple years, 1 to 2 years. Coho salmon spend 1 to 3 or sometimes even 4 years. Sockeye salmon spend a couple years. So it just depends on the species. Coho salmon are very variable as a species. And so down where you are, I think they just spend like 2 years. Up here, they spend longer. The habitat we have a very plentiful habitat here. So that's, probably one reason they can grow. As Jacob mentioned, they hatch out. They're about 30 millimeters. So they're, like this big when they hatch out. And then they grow each year. And so we can tell kind of by growth, because we've caught so many, how big they are. But in their first year, they're going to get to be, I don't know, about 50 or 60 maybe millimeters. By their second year, they're 70, 80, depending on how rich the habitat is where they are. And then, and you'll see this a little later on, they're about 100 millimeters when they're ready to go out to the ocean. So like this big.
Quite a bit of difference, yeah. So is that the biggest salmon that you get, or is the 100 millimeter ones?
Well, no, we do get ones that are 130, 140. We get all excited. They're big fatties. There's just a lot of range, just like I'm a small person. I'm 5 foot tall. Jacob's 6'1". So there's a lot of range in people too. It's just we're different. Salmon are different sizes, but they're yeah. I'm not sure why some of them get so big, but in general, the bigger they are when they go out to the ocean, the better off they will be. The more likely they are to survive.
Yeah. Well, I see that we have about 11 minutes left. I was wondering if you could give a really short answer to what do you do with non-salmon species of fish that are caught? Do you just let them go, or do you take information on them too?
We do take information because we want to know the community that the salmon are living in. Usually we take species and length data from them, and of course numbers. So we keep track of numbers, number, species, length. Sometimes we try and pit tag them, which sometimes we pit tag a sculpin or something like that. But mostly we're focused on the salmon for those types of studies. Oh, we take diet data from them too. What are they eating? These other species.
Yeah, it's good to get information from whatever fish you can get, I think, to help with that. I did have one more quick question before we go into your last section, because this one's from Facebook, from Merle Hawkins. They said that they had major flooding on our Kachemak Creek with evacuation of residents due to danger of dam breach. And they were wondering, will this have affected our mostly pink salmon eggs? And they also have a salmon hatchery by Sarah. So Coowe, you've frozen a little bit.
That's a really good question that I. Jacob, do you want to try? Can you guys hear me now?
Oh. I heard you for a second there, Coowe. I think what Coowe is alluding to is it is a really interesting question. It'd be hard to say, I think, for me personally, just because the southeast systems are so different from what we have over here. They're a lot steeper, a lot faster moving water. But I would imagine if there's a large input of, say, mud and debris and stuff, it probably is going to damage whatever juvenile fish are in those streams at that time.
Can you guys hear me now?
Yes, we can hear you now. So
okay.
Maybe we should move on to our last section. Oh, sure, go ahead.
Okay, I just wanted to say one thing about that. It's a really good question about what the effects of flooding. Salmon in general are really good as a group of dealing with disasters. They've evolved in situations where there's volcanoes and earthquakes and mudslides. And so while many of the fish may die, there will be some that survive, and that will keep the species going. So salmon are really good at basically dealing with disasters if we give them, the opportunity. Okay, so let's move on because we are running out of time, and I really want to share the next group of slides with you, which are all about what salmon are eating and doing in the winter. Oh, but before that, let's go to the next slide. And
so do you want the one after the salmon showing in the little container here?
Let's see. I am not seeing the slides right now, so you might have to tell me. I think my internet.
Oh, sorry. That's because I haven't shared my screen. Duh. Okay, so here we go.
There we go. Okay, yeah, let's just go here. So here's Sabrina holding some salmon and just showing you that, yay, salmon in winter. And but what happens if you can't see the water? Like this is an open water area. So if you go to the next slide, and this is one of our colleagues named Dan, and you can see here we had to dig a hole, and you can see how thick the ice is. If you go to the next slide, here's another couple of technicians working with us. Dustin there on the left is 6 feet tall, and we got Tim on the right, and we were just having a fun competition. Gentlemen, start your engines, because we had to dig a lot of holes to get in order to set minnow traps. So if you go to the next slide, you can see here's Dustin, and remember he's 6 feet tall. He got that hole started, but we still had to scoop out a bit. It's a lot of hard work to get a bunch of minnow traps in when you don't have an open water lead. And if you go to the next slide, and here you can see all these holes with the promise of fish. Those are the strings from the minnow traps up above, and we leave them overnight and come back. And often we had to re-dig them out because it froze so much overnight, but not quite as hard as it was the first time. And then if you go to the next slide, and success, yay. So here we get to, we pull our traps out, and we cut the baby fish even under the ice. So yeah, and that's a whole nother realm of conversation, which we don't have time to get into here, but maybe if anybody has questions, they can contact me or Jacob with questions about fish under the ice. Okay, so let's move on to what they eat, because this is really cool. You guys are going to like this. And how do we know what they eat? Well, we do something called gastric lavage. Here you can see a fish who's been anesthetized, and I'm pumping that fish with water through this syringe into its stomach. So the syringe is in its stomach, and I'm squeezing water in, and that water then forces what's in the stomach to come out. And if you go to the next slide, here we are, and you can see that fish is being lavaged I like to joke that it's the fancy French word for making a fish barf, because gastric is stomach and lavage means water barfing. So here's that fish being lavaged, and we have a funnel, and then whatever's in the stomach ends up in that plastic bag there, and that's our sample. If you go to the next slide, and yes, our hands do get cold. So what do the fish eat? This is a caddisfly, so we're trying to figure out, like, what do they eat in the winter and what do they eat in the summer, and is there enough food? Are they growing? If you go to the next slide, it's a little cartoon just to get across the point that salmon are cold-blooded. So in our systems, there's plenty of food for them, but because they're cold-blooded, all their metabolism slows down, and even though they might have full stomachs, they can't actually digest it very quickly because their ability to all their physical processes are slowed down by the temperature. If you go to the next slide, we want to know, one of the things we're doing is we want to know if they're growing in the wintertime. And so this is a scale. You can see the fish is in a little bit of water on that scale. And we can go back, and because they're tagged, we can repeatedly measure the same fish over and over and see if they're growing. And in fact, we find that most of the time they are not growing during the winter. In fact, they lose weight often over the winter, but they regain it really quickly in the spring, and they grow really quickly. And if you go to the next slide, they grow really quickly, and then some of them end up going out to the ocean. Not all of them, but some of them end up going out to the ocean. And we talked earlier about the size they were when they go out to the ocean. And here's a little question for you. I don't know if we have time, Lisa, about, how big do you think they are? If you remember from we were talking about this a little bit earlier, like, how big do you think they are roughly when they go out to the ocean? Are they the size of a paper clip?
Yep. So why don't you type in really quickly whether you think that they're paperclip, pencil, or pad size. Texas thinks that they're pencil size. A couple other people are thinking they're paperclip. Nobody seems to think that they are the notepad size. So what do you think, Coowe?
That's right. Pencil would be the closest, I think. Sometimes they go out as a very small fish, and that's a whole nother type of life history. But in general, they're about this big, a short pencil, carpenter's pencil. So when they leave, they smoltify, and I think I used that word earlier, and that just means that they use the estuary, which has both salt and fresh water, to adjust to the conditions of the ocean. And they generally turn silver. So here you see this Coho salmon, which has lost its par marks, those black marks on its side. It's starting to look more streamlined. It's generally becoming more of what you would find in the ocean, a silver fish. And they adjust not just their color, but also their metabolism. If you go to the next slide, please. So this is basically bring us back to the big picture. We've been talking about the anadromous life of a salmon, and particularly focused on the juvenile salmon, and thinking about studying them in the summer, but also really studying them in the winter, which is, I hope you learn to appreciate that it's hard to do. It's hard work, but it's really important because it's a critical time of their lives. It's a time that they really need to survive, and sometimes over multiple years. It's kind of like what we call a bottleneck. So it's a really critical time that they need to survive in order to move on to the next stage in their life, and it's a tough time for them. So it's important for us, and if you move on to the next slide, is kind of to remember that when we're out and about in the winter here in Alaska, if you're in Alaska or in a place where there are juvenile salmon, that there are salmon under the ice, and you may not know they're there. So, just thinking about when you're driving your ATVs or things like that, just remember that, there could be baby salmon living in your backyard.
Well, great. I'm really glad that we were able to show some of those pictures of you guys doing winter research, because I think that's one of the really interesting work that you guys do in terms of finding out what that the fish aren't actually growing that much. Ola did have a question of what is the winter diet compared to the summer diet?
So they're pretty. They eat a lot of different things. The food they eat similar foods, except I want to point out one thing that's so they're eating basically caddisflies, a lot of midges, chironomids we call them, but those are like the non-biting midges that kind of annoy you when you're down by a stream. Caddisflies, mayflies, stoneflies. But here's a really interesting thing we found. In the spring, they start gorging on worms, earthworms, which actually aren't native here. They've been here for a long time, but they're brought in by people pretty much. And they eat so many of these worms that you can actually, let's see if I can, you can see like the outline, like my finger, you can see the outline through their skin of the worms. Their stomachs are so full. And they do it within a very short period of time when the spring is thawing and when the ice is thawing in the spring. And they gain weight super quickly. It's very remarkable, and it's something that we want to explore more. We've seen it in several different locations and even with different species, so Dolly Varden as well as Coho salmon, gorging on these worms and rapidly increasing their weight so that they can move out of the winter habitats.
Wow, that's a great image to end on. So we are out of time, but I really wanted to thank you very much, Coowe and Jacob, for coming and talking with us about salmon under the ice. We learned, I think, a lot about winter habitat and how you go study salmon in the winter. And thank you to all of our audience members for coming on this week and listening to us. Your questions are great, and I really like how engaged you are with our webinars. So thanks very much, and tune in next week. We'll have Deanna Krauser talking about zooplankton, so we'll find out a little bit about the food web in the ocean. But I really appreciate everybody's time. Thanks very much.
Thank you all.
Thanks.